recording so I don't forget that. Got one more minute. We'll let people trickle in before we get started. All right, one more minute. We are going to start on time because we got lots to cover, and I don't want to feel rushed, but at the same time, I do want to make sure we cover everything. Hope everyone had a great weekend. Weather's been fairly nice. Sun's been shining. Has it been too cold? Has it been hot? Check. Okay, yeah, the clock on my computer says that it is indeed 11:30, so we're going to go ahead and get started. So let me share my screen, and we'll just jump in with 2 feet here. Okay, well, I want to start with a couple of announcements. So we're going to be finishing the chapter on blood today, and so this afternoon, your quiz on blood will open, and it will be due on Wednesday at midnight. Setup will be the same as the muscular system and the endocrine system. 10 questions taken from a pool, 9 minutes to complete them, unlimited attempts, and your highest score counts. So it's another what's called a quiz for mastery. Also, I've got to go into Blackboard and add your quiz on the endocrine system to your score. That has not been done yet. It's not going to change your score that much, but I haven't done that yet. It has been graded. The computer automatically grades it, but I just realized I have not added it yet. Your practical has been graded. I did send you an email on that. You should be able to see your score. Your current points and your grade does reflect your practical. However, I'm withholding your ability to see the actual practical in terms of the questions that were asked, your answers, and the correct answers until I have a couple of makeups due. I might be able to shield it from people who haven't taken it yet while allowing people who have taken it to see it, but I'm not sure, and I'm afraid of opening it up and letting students who haven't taken it see the exam. I don't think that's how it works, but I just want to make sure before I open it up for everyone to see. But at the latest, by this time next week, you'll be able to go in and actually see your practical, see what you got wrong, and see where if the computer graded it wrong, but then I went in and overrode the computer and gave you credit where that occurred. So we are about to finish the last chapter before your exam. So your exam is scheduled for next week. It will open on Thursday, April 30th, and it will close at midnight on Friday. Let me stress that even though you have an exam that you have to complete, we will be having class on Monday, Wednesday, and Friday of next week, just like we normally do. I have to have that time because we are a little behind. That exam will cover the muscular system, the endocrine system, blood, and the cardiovascular system. It will be all true, false, and multiple choice questions. I'm not going to have the short answer, and I'm not going to have the matching, because what I found with another class is it's just too easy to cheat. It's just too easy to Google answers. I'll try to have a better breakdown of that prior to the exam next week. There have been questions of take-home exam, so I'm going to push the deadline for the take-home exam back until May 8th. So I'm going to post the take-home exam online so you can start putting in your answers if you so desire. They just have to be in and submitted by May 8th. Now, the take-home exam and the key are on campus, and I got permission to go on campus tomorrow to retrieve that. And that's why I haven't posted them online yet. Once I do that, you'll have the ability to start entering your answers if you so desire. So you can enter them like a piece at a time and then save don't submit. Then come back and add some more and add some more and add some more. And then when you're like, okay, this is final, I'm finalized, I have everything exactly what I want, then submit. I'll talk more in detail about that once I have posted that exam online and you can start inputting your answers. Okay, any questions about that before we jump back into the content? I know I went through quite a bit. About 10 minutes ago or so, I emailed all this information to you. I did forget to mention the take-home exam, and a student brought that to my attention. So thank you very much for doing that. So I included it in these announcements. Questions? Don't be shy. Okay, yes.
How much time do we have for the exam online?
The exam online, let's see, let's assume if it's 50 questions, then it will, gosh, you know, I gotta really look at that. I think it was 42. I think it's 42. I'll provide more information.
So kind of like the lab was?
Correct.
Okay.
What I allow for is about 0.8 minutes per question. Let me see what but before I finalize that, let me see what I did with the other class, because that seemed to work well.
Sure.
And let me get back to you on that. So I'll make a note on that. Thank you. The finals on the syllabus. Let's see here. What I have it. I have it for Wednesday the 20th. It'll open Wednesday the 20th. It will close Friday the 22nd at midnight. Okay, other questions? Questions going once? The true and false questions will just be true and false. There will not be any replace the word that's wrong, because I can't do that electronically. So they'll just be true and false, flat out. Okay, so any other questions you have, if you could hold them off till the end of class today, so we can go ahead and get started on the content so we don't fall any further behind. Thank you very much for your questions. I encourage them. Okay, so so this is where we left off on Friday. We ended our discussion on hemostasis, which is the stoppage of blood. And so the next topic has to do with blood group, blood typing. So we're going to explain the ABO and RH blood groups. We're going to explain how blood is typed for the ABO and RH blood groups. We're going to explain what causes the rejection of donor blood by a recipient. Then we're going to talk about the hemolytic disease of the newborn, what that is, what causes it, and we're going to describe how the site of hematopoiesis, that's blood cell formation, changes in the fetus during pregnancy. Okay, so even though the body has mechanisms to stop blood, in some cases the damage is so extensive that there's excessive loss of blood. Now we can lose about 15 to 30% of our blood without that becoming life-threatening. We may be weak, you know, and we may not feel quite ourselves. We won't die. However, above 30% and blood volume drops so low that it's not possible for the cardiovascular system to maintain blood pressure, and therefore can't provide all the vital organs and tissues of the body with sufficient blood flow. And so what happens is the person begins to go into what's called shock, as organs are deprived of blood flow and oxygen, and they begin to die. At that point, the only way to treat a patient is through a blood transfusion, and that's the only way to really quickly raise the blood volume of a patient. And of course, it's important to type both the donor blood and the recipient blood because if the donor and the recipient are not compatible, then you can have a rejection of that blood transfusion. So let's talk a little bit about what's involved in that. So I don't know if you recall, but back when we talked about the cell, specifically the plasma membrane, I mentioned that there were glycoproteins, that's a protein plus a small carbohydrate unit that's found on the outer surface of the plasma membrane of cells, and those are antigens. Your immune cells sort of use them to identify cells that belong to the body, what are called self, and contrast those with cells that are foreign. If you ever wondered, well, how is it that your immune system can recognize and not attack your own cells, but recognize foreign cells? Well, it's because of these glycoproteins or these antigens. Self antigens are not attacked, or at least normally not attacked by the immune system, and foreign antigens are. And the red blood cell, like any other cell in your body, has these little antigens. You can think of these antigens as little hello, my name is stickers that your cell uses to recognize self from foreign, or your immune system rather. Okay. Now, red blood cells have over 30 common antigens. However, there are only two groups of antigens that are usually responsible for the most vigorous transfusion reactions. It's not that they're the only ones, but they're usually the ones that are involved in blood transfusion rejection. So those are typically the only two that are typed, and they are the ABO blood group and the RH blood group, and those are the two that we're going to discuss now. So the ABO blood group is based on the presence or absence of two antigens. You have an A antigen and you have a B antigen. If the A antigen is present, and only the A antigen, you have type A blood. If the B antigen is present, you have type B blood. If you have the A and B antigens, then you have type AB blood. And if you have neither antigen, then you have type O blood. Give me just a second. My son's being a little louder than I'd like him to be. I don't know if you can hear it, but I can hear it. It's disturbing me. Forgive me. So give me just 1 second. Okay, I apologize for that. All right, I don't know how many of you know your blood types are, but I'm type A, so my cells have the A antigen. Okay, so this is a part of a figure from your book, and it's just sort of illustrating this, right? So I'm type A, so my cells have these A antigens. If you're B, you have these B antigens. If you're AB, you have both A and B, and if you are O, you have neither. Now, if you have what are called antibodies against whatever antigens you don't have, so your immune system produces these proteins called antibodies, and they will attack the antigens that your red blood cells don't express or don't have. So, as I said, I'm A. So in my plasma are anti-B antibodies. These are antibodies that will attack the antigens, the B antigens on a red blood cell and destroy the cell. If you are B, you have anti-A antibodies. If you are AB, you don't have antibodies against either one, because if you are AB and you have the anti-A antibodies, your immune system would destroy its own red blood cells. Likewise, if you had the anti-A antibodies, the same thing would happen. So someone who is AB, they don't have antibodies against either A antigen or B antigen. If you are O, you have anti-A and anti-B antibodies. It is the presence of antibodies that determines what kind of blood you can receive. If you have the antibodies against the antigen, you can't receive donor blood that has that antigen. So someone who is A can receive A blood, but they can't receive B blood because they have anti-B antibodies. Can they receive O blood? Yes, they can, because there are no antigens on the red blood cell for the anti-B antibodies to attack. Someone who is AB can receive A blood, B blood, and O blood because it has no antibodies against the A or B antigen, and of course, O has no A or B antigens at all. Okay. Now, here's the interesting thing. Usually, we don't produce antibodies unless we are exposed to an antigen. So in other words, usually if you have antibodies floating around in your body against a virus or a bacteria, it means you've been exposed to that virus or bacteria. That's the whole that's how vaccinations work. You are exposed to the antigen of whatever virus or bacteria causes the disease. Your body then develops antibodies so that if you're ever really exposed to the real thing that could cause you to become sick, your antibodies clear out that virus or that bacteria before you get sick. However, with the blood groups, what's interesting is you develop antibodies against whatever antigen you don't have without being exposed to it. Now, from what I've read, it may be that in some of the meats that we eat, even baby food, that there's blood with antigen in it that comes from that, or it could be that there's bacteria in our gut that produce an antigen that's similar to the A and B antigen, and so you develop it that way. Not really sure, but several months after you're born, you develop antibodies against whatever antigen you don't produce. I'm not ignoring you, Dawn. I'm just going to get to a stopping point and then try to address your question. Okay, so rejection occurs when a patient is transfused with an incompatible blood type. And what happens during that process is the red blood cells are destroyed. And the whole idea of having the red blood cells is to carry oxygen. So if you get a transfusion of blood and then the erythrocytes are destroyed, it completely negates the benefit of that transfusion. Also, the hemoglobin released from those erythrocytes can actually damage the kidney and cause kidney failure. So it can make a bad situation even worse. So that's why we worry about the rejection of a transfusion. So that's the ABO blood group. Let's talk about the RH blood group. So there's one of 8 RH antigens, and this it's called the agglutinogen D, believe it or not. But I guess the agglutinogen D is too many syllables and too hard to pronounce. So we just call it the RH factor. You may have heard it called that, and it gets its name because agglutinogen D was identified first in rhesus monkeys, and rhesus monkeys begins with RH, so that's where we get the RH factor. And the situation is, with respect to this blood group, either you have the protein or you don't. If you have the protein, then you are considered RH positive. If you don't have the protein, you are RH negative. So I am A positive. I have A antigen and I have this agglutinogen D. Most Americans are Rh positive, but certainly not all. Now, the Rh factor, or Rh, the agglutinogen D, with respect to production of antibodies, follows the same pattern we find with most antigens. You don't produce the antibodies unless you're exposed. For example, if let's say one of you is A negative and you needed a blood transfusion, well, assuming you hadn't ever been exposed to positive blood before, you could actually get a blood transfusion from me, even though I am A positive and you are A negative. Because you've never been exposed to positive blood, you wouldn't have antibodies floating around in your system. Now, after you got a blood transfusion from me, well, then you would, because you would be sensitized. That's generally how the production of antibodies work. However, the danger that can occur has to do with pregnancy. If you have an Rh positive father and an Rh negative mother, as shown here. So let's say, and this was a situation with my wife and I, believe it or not. So my wife is Rh negative. I can't remember her ABO blood group. So she's Rh negative. I am Rh positive. That means that we have a 50 to 100% chance of having a child that is positive. Just depends on if I have 2 Rh positive genes or 1, but there's at least a 50% chance. Now, during pregnancy, fetal blood and maternal blood do not mix. So there is no issue with the first pregnancy. There's the the Rh negative mothers not producing antibodies against the Rh positive antigen because she's not exposed. However, during the birthing process, which is very messy, let me tell you, you have the rupturing of the well, we have the cutting of the placenta, but that's not what I'm trying to get at. You have the maternal and the fetal blood supply in very close proximity in the uterus here, right? And so during pregnancy, what typically happens, during parturition, during the birthing process, right, you typically will have because this detaches from the walls of the uterus, you will have blood mixing. And so at the time of birth, you will have the mother being sensitized against the Rh protein, but the baby's already born. There's not that, there's no issue. Okay, this is the placenta, by the way, right, where you have the umbilical cord connected to the child or the baby, and then you have the fetal blood supply in close proximity, but not mixing with the maternal blood supply. During parturition or birthing process, there is, this is very messy as it's detaching, and you do have the mixing. And at that point, the mother can be exposed to the Rh protein and can be sensitized against the Rh protein. So then, second pregnancy comes along, and if we have a Rh positive child, and we have a mother that was sensitized against the Rh protein from the previous pregnancy, that can cause a problem, as antibodies could cross. Even though the blood doesn't mix, those antibodies are small enough, they can cross the placental barrier and enter the fetal blood circulation and attack the red blood cells of the fetus. And if that happens, if it's severe enough, you can get what's called hemolytic disease of the newborn. So if you have an Rh negative mother, what they will typically do at several times during pregnancy is give her a injection of what's called RhoGAM, and that blocks the sensitization. However, it's not 100%. My wife was sensitized during the first pregnancy. She got a shot of RhoGAM, I think it was at 3 months, and also at parturition, and she still got sensitized. So the second pregnancy, we had above average normal sonograms and visits with the pediatrician. There were no issues, right? But it was just precautionary, and it was considered a high-risk pregnancy because of that. Okay, so obviously it is important that donor blood and the recipient's blood is typed. If we're looking at a blood transfusion, and also if we have a mother that, an expectant mother who is pregnant. So let's talk about how that is done, okay? So I mentioned that antibodies will attach themselves to antigen, and they'll do that, and they'll destroy the red blood cells. These antibodies are very specific to their antigen, so they don't attach to anything else typically other than their antigens. So in a lab, we can use them as a probe. We can use them to determine, if we have a blood sample, if it has a specific antigen. So if a lab were to take my blood and they didn't know what my blood type is, they could take a little sample of my blood and they could mix it with a solution that had anti-A antibodies. And what would happen is those anti-A antibodies would stick to the antigen of my red blood cells, and it would clump like what's shown here. And you can actually see this with the naked eye. You don't have to look under the microscope. You can actually see this with the naked eye. And that would tell them that, hey, his blood has A antigen. So either he's A or he's AB. So then if they want to determine whether or not I'm A or AB, they could take another sample of my blood and mix it with the anti-B serum. And if it didn't clump, then it would mean I'm A. If it did clump, it would mean I am AB. And then they do the same thing with the Rh antigen and antibodies to determine if I were positive or negative. So let's read these tests that were done in a lab. So we've got 4 different blood types here. And as I said, the way you type blood is you use anti-A and anti-B antibodies, and you mix them with someone's blood, and then you look for the clumping. If it clumps, it means that the antibodies found antigen. If you don't have clumping, it means that the antibodies didn't find their antigen. So in this first sample here at the top, we took a blood sample, we mixed it with anti-A serum, we got clumping. We also mixed it with anti-B serum, and we got clumping. So this person is what? They are AB. That's their ABO blood group. We used those antibodies like probes, and we found anti-A antibodies. Sorry, we found the A antigen, and we found the B antigen. If we took let's take a sample of blood from another patient, and we're going to mix it with anti-A serum and anti-B serum. Notice we don't get clumping with the anti-A serum, so there are no A antigens there. We do get clumping with the anti-B serum. And so this person is what? B. Take a sample from a third patient, we get clumping with the A, we don't get clumping with the B, so they're A, that would be me. And if we don't get clumping with either the anti-A serum or the anti-B serum, it means that there's no A or B antigen there, and so the person is O. That's how blood is typed. And again, this is with the naked eye. You can actually see this. It looked just like this with the naked eye. Now I want to share with you a website that has the coolest game. That is not the website. The coolest game that you will ever see. Educational game, that is. Because now usually educational games are sort of eh. This one is awesome. This is the best educational game I've ever seen. It's called the Nobel blood typing game. And if it pops up here, I have a link to this from Blackboard. So we have a patient here that needs blood. First thing we got to do is we got to type the patient's blood. So we take a sample. We have our A, B, and RH anti-serum here. So we're going to drop blood in each of those. And we're going to look for the clumping. Right? If we find clumping, we don't have any clumping here. The anti-B, anti-A antibodies don't find antigen. So no clumping here. Again, the anti-B hasn't found any antigen, right? So what's the blood group? O. No A antigen, no B antigen. What about RH? We got clumping. And if we looked under the microscope, we'd see that our anti-RH antibodies have found the RH antigen. So this person is O, and they are O positive because the antigen was found. So now that we know they are positive, we're bloody right. Now that we know they're O positive, now we can give them blood. So if they're O positive, so they need 2 units of blood. If they're O positive, certainly we can give them O positive blood, right? Right? Can we give them B positive blood? Let's see. Oh, nope, guess we can't, right? We can't give them B positive blood because they have antibodies against the B antigen. Can we give them O negative blood? Certainly we can. Well, they're O positive, so we can give them O blood, and we can give them O negative blood because, well, they have the RH antigen. They don't have antibodies against the RH factor, and this doesn't have the RH factor anyway. So we can give them O negative.
Yeah.
Anybody can get O negative blood, by the way. That's what we call universal donors. Okay, so this is a really, really cool game. I encourage you to play it. It's educational and it's fun, to be honest. Okay, so let's go back to our slots. So as I mentioned already, RH factor typing is done the same way. You would just mix the blood sample with the anti-RH antibodies. Now, something else that's also done is, in addition to typing the blood, even after you say, hey, we got a recipient that's A positive and we got a donor that's A positive, they still, before they send that blood out to be transfused, they will mix the blood, little samples of the blood together. It's what's called cross-matching. So if you've ever heard some of the medical shows type and cross-match, they also mix the blood even after they've typed it. And why might you think they might do that? Any thoughts on that? Well, remember I mentioned that there are 30 different antigens. There's 2 groups that are usually responsible for any significant rejections, but not always. So we want to mix the bloods, even if they have the same ABO and RH bloods, just to make sure that the other blood groups or antigens are compatible. Okay, so Don, you had a question. You said, what's the point of antigens if people with O blood get along just fine? Could you clarify that question? Don, are you there? Okay, well, you can always save it till the end. All right. So why have any glycoproteins at all? So again, your immune system uses those glycoproteins to determine, self from foreign. However, I am fairly certain that the body did not necessarily anticipate getting donations from other humans. It's more geared toward microorganisms and things that can hurt you. So there wasn't, the design was not necessarily protect you from other humans. It was designed to protect you from microorganisms, which could cause you harm. The the fact that some people can get blood from a greater number of humans versus others is incidental. It's not by design. To my knowledge, I don't know of any increased risk of any disease related to blood type. Now, me not having knowledge of that doesn't mean that that exists. Okay. But to my knowledge, there's no significant difference in risk of disease among the different blood types. There is some anecdotal evidence, interesting enough, that COVID-19, that people with type A blood, such as myself, might be at increased risk. But again, this is very preliminary observations, and and to my knowledge, no one knows the why. It's just something that what they did notice in China was there is a slightly higher a higher percentage of people with type A had COVID-19 and were sick versus the population as a whole. So it's something that they're going to be looking at. Okay, I want to finish off the chapter on blood by talking about developmental aspects of blood. So in the fetus, the liver and the spleen and some other sites initially make blood, and then by the 7th month of gestation, the bone marrow takes over. Also, fetal hemoglobin differs from the hemoglobin that's produced postpartum, that is after birthing, and that's because the fetal hemoglobin binds to oxygen more tightly than adult hemoglobin, and that's so you get the transfer of oxygen from the hemoglobin of the maternal hemoglobin to the fetal hemoglobin. Okay, and that ends our chapter on blood. Okay, I just want to make sure that, okay, Michelle.
So I had a friend that a couple of months ago had a C-section, and they cut her placenta in the process, and the baby had to have blood transfusions and was in the NICU for a couple of weeks.
Okay.
Would the mom have been Rh positive, or is that just precaution that they do something like that?
Well, so I don't have enough information really to speculate. Was it a planned C-section?
Yeah, it was a planned C-section and a tubal after baby was born, but I think she had said like her placenta was in a weird spot, and they ended up cutting that as well during the C-section.
Oh, okay. So my guess is that the baby lost a significant amount of blood, and they had to transfuse the child just to replace the blood that was lost. Because until the umbilical cord was cut, there would be blood flowing through that umbilical cord. And if they cut the umbilical cord they cut the placenta, then depending on where it was cut, there would have basically the fetal circulation of blood would have been going through that, and the child would have lost blood there.
Okay, makes sense. Thank you.
That's what it sounds like anyway. Okay, other questions? Okay, so as I mentioned, we are there is going to be a quiz posted this afternoon on the chapter on blood, and it will close on Wednesday at midnight. So we're going to move on to the next chapter, which is very much related to blood. It is the cardiovascular system. This is chapter 11. Soko, did you have a question?
Hi.
Yes, do you have a question?
Yeah, I typed it out, but I was wondering if people with sickle cell-shaped blood cells can receive transfusions okay, or if there's trouble with that.
To my knowledge, there's no problem with receiving a blood transfusion. I'm certain they would not be good candidates to donate blood, but I don't see any reason why, if they have compatible blood in terms of the ABO and RH blood group, where that would be a problem. The issue with someone with sickle cell anemia, I'm assuming you mean sickle cell anemia, is with the hemoglobin molecule, not these antigens that are expressed on the plasma membrane. All right, let's jump into the cardiovascular system with 2 feet in the 19 minutes that I have left. So the function of the cardiovascular system, I always start with the function because everything else we talk about is going to relate to that. We're going to start with the function of the cardiovascular system and list the components of the cardiovascular system. So the function of the cardiovascular system is what? Transportation, right? The cardiovascular system circulates blood throughout the body. It uses blood as the vehicle to transport nutrients, respiratory gases, hormones, cellular wastes, heat, and other substances, such as white blood cells. Remember that cells can only interact with their immediate surrounding. Think of the cells in your pinky toe. They can't run up to your small intestine when they need nutrients. They can't run to your kidneys to get rid of wastes. They can't run to the various organs of your body. Sorry, not organs. They can't run to your lungs to get oxygen. They can't run to the various endocrine glands to get their daily dose of hormones. All that has to be brought to them. And waste products and things that they secrete have to be taken away from them. And that is the function of the cardiovascular system. It's going to continually refresh this interstitial fluid that bathes these cells. So cardiovascular system's function is bring to the cell everything it needs, remove from the cell all the waste products or anything that cell produces that's needed by other cells. So it transports nutrients and waste products and respiratory gases such as carbon dioxide and oxygen, hormones. The cardiovascular system is composed of your heart, which is the cardio part, and vascular, which refers to the blood vessels. The heart creates a pressure gradient. Remember, gradients cause movement. The heart creates a pressure gradient, and blood simply moves from high pressure to low pressure, and the blood vessels provide that pathway. So blood is the vehicle, the blood vessels are the pathway. So you can think of like the blood's like the car. The blood vessels are like the interstate, the Eisenhower interstate system of highways. And I guess the gas would be like the heart. So the heart would be like the gas. Okay. Sometimes, people use cardiovascular system and circulatory system interchangeably. Technically, there is a difference. Cardiovascular system is just heart and blood vessels. Circulatory system is heart, blood vessels, and blood. For the purposes of this class, you are fine to use those terms interchangeably. So blood vessels allow the movement of blood between the heart and other tissues of the body. Couple of definitions. Arteries carry blood from the heart. Veins deliver blood back to the heart. Forget what you were told about, well, arteries carry oxygenated blood and they're always red, and veins carry deoxygenated blood and they're always color-coded blue. That is mostly true. Usually, most arteries carry oxygenated blood and they're color-coded red. Most veins carry deoxygenated blood and they're color-coded blue. But those are not the definitions. Arteries carry blood away from the heart. Veins bring them back. Some arteries carry deoxygenated blood. Some veins carry oxygenated blood, as you'll see in lab this week. Capillaries are the blood vessels where there is an exchange of substances between the blood and the tissues. The arteries and veins are just moving the blood from the heart to the tissues, or to the tissues back to the heart. In the capillaries is where there's an actual exchange of substances like oxygen and nutrients and hormones and etc. between the blood and the tissues. Please understand that the cardiovascular system is a closed system. I know every time we are just scraped, we bleed. And it may seem like blood is everywhere in the body, but it's not. It's confined to our blood vessels, right? The cardiovascular system is a closed system. We're going to start with talking about the heart. Hopefully I'll get through at least these objectives today and we can end on time, and we'll pick up from here on Wednesday. So we're going to describe the location of the heart. We're going to describe the function of the heart. We're going to explain the function of the coronary circulation. And then I'm going to define a few terms: ischemia, angina pectoris, and an infarct. And that should be an R, infarct, not an in fact. So this is an illustration of the thoracic cavity, which is where your heart is found. And your heart is actually found in a cavity within the cavity. So the central cavity here within the thoracic cavity is called the mediastinum. It's where your heart, these great vessels, and though not shown on here, your trachea and esophagus are found here as well. This is called the mediastinum. And on the left here, you see a cross-section of the thoracic cavity, and you see a cross-section of the mediastinum there. Your heart is slightly left of center. It's not right through the bottom. It's slightly left of center. This region here, which is right about where your second rib is located, this region here, and this is a region, it's not a structure, it's a region or area, is called the base. It's where the great vessels emerge from the heart. And then located in the inferior portion of the heart, on the left side is this little point, and this is called the apex. The heart sits on the diaphragm, which separates your thoracic cavity from the abdominal cavity, and the apex of the heart here points toward the your left hip. Now, it may sound odd that this top portion is considered the base and the lower portion is considered the apex, because typically we think of the base at the bottom and the apex at the top. Well, a base is not what's at the bottom. By definition, it's the point of attachment. Because of gravity, typically the base of something is at the bottom. However, the heart is attached to the body through these great vessels. So this is considered the base. And your apex is always opposite your base. So that's why this portion here is considered the apex. Your heart is an amazing organ. It pumps blood to all of the tissues of the body. And it does it 24/7, whether you're awake or you're asleep. And it moves about your entire blood volume, which is roughly 5 liters every minute. right? And it never stops. While you sleep, your heart moves blood equal to 30 times its own weight, right? If the heart moves 5 liters a minute, just think about this: 5 liters times 60 minutes times 24 hours, right? So in a 24-hour period, that heart's going to move 7,200 liters of blood, right? And then just multiply that by 7 days, right? That's 50,400 liters of blood. I just find that just amazing, okay? Blood that moves through the heart, is not actually used by the heart as a source of nutrition. It's moving too quickly. So the heart has its own what's called coronary circulation that it utilizes as a that it uses to get the blood it needs. The heart has 2 what are called coronary arteries that come off the aorta. They bring blood, oxygenated blood to the heart, and then it drains back into the heart. Well, actually, you know what? We are at 12:20, so we'll stop right here, and we'll pick up right here on coronary circulation on Wednesday. Something I did not mention, which I had meant to, and that is everything that you need for this week's lab, which is on the cardiovascular system. I'm sorry, it's on the cardiovascular system, but specifically it's on the anatomy of the heart and the anatomy of the blood vessels. Everything that you need is on Blackboard. The pre-labs are there. The presentations by me are there. And the Quizlets have been posted. So go ahead and jump in and get going on that. It's one lab, but I've separated the anatomy of the heart and the anatomy of blood vessels. So you have a pre-lab for anatomy of the heart, you have a presentation for the anatomy of the heart, and you have a Quizlet for anatomy of the heart. Likewise, you have a pre-lab on blood vessels, you have a presentation on blood vessels, and you have a Quizlet on blood vessels. At one time, they were 2 separate labs, but they hacked off a week of our classes a few years ago, and they went from 16 weeks to 15 weeks, and so we had to reduce and combine those 2. But I still sort of keep them separate because that's how the lab manual is sort of organized, and it makes it easy to do that. Okay, so I'm ending our formal class now. I'm going to stop the recording, and if anyone has any